I think this is a good place to look a little bit at the genetic mix that we carry as the only remaining Homo lineage. So that's the Homo sapiens, and obviously we will, as we go along, we'll revisit some of these things, but also we will talk about the human diaspora, which we talked already about, and despite all the uncertainties, genetic methods, especially using models that assume mutation rates and so on, the so-called molecular clock approach, without getting into details, that allows the genetic approaches to figure out what are the various mixes that must have happened along the way. Okay, so this paper in Nature looks at the genetic mix and how it may have come about. We identify three key phases that are surrounded by major questions and which will be at the frontiers of future research. The most recent phase comprises the worldwide expansion of modern humans between 40 and 60,000 years ago, and their last known contacts with the archaic groups such as Neanderthals and Denisovans. We'll keep coming back to these terms and the archaeological sites and so on beyond what we have already seen, especially in terms of how climate change has occurred over these places and how that might have affected diaspora and speciations and maybe diets and so on. The second phase is associated with a broadly construed African origin of modern human diversity between 60 and 300 kilo years. So if Homo sapiens arrived somewhere around 300 to 200 kilo years ago, and we had many other hominin species coexisting and slowly disappearing over time. And by 60 kilo years ago, we only had maybe Neanderthals, sapiens, Denisovans, and Floresiensis. The oldest phase comprises the complex separation of I have some noise here, sorry. Okay, the oldest phase comprises the complex separation of modern human ancestors from archaic human groups from 0.3 to 1 million years ago. We argue that no specific point in time can currently be identified at which modern human ancestry was confined to a limited birthplace, and that patterns of first appearance of anatomical or behavioral traits that are used to define Homo sapiens are consistent with a range of evolutionary histories. So obviously we'll keep dealing with uncertainties in climate and their impacts on anatomical, behavioral, and adaptation, and so on and so forth. But here the focus is mostly just on the genetics, and I'm going to be brief as usual because you can read up more if you are inclined to follow this up. Okay, so we start with phase 3 and then 2 and 1. So looking at the modern human-like remains or material culture from archaeological sites here, you can see that these are older here. The Fuyan cave in China, Zeren, these are around 100 kilo years. This is Lida Her, around 68 kilo years, and Majé Bebe, however it is pronounced, around 65 kilo years. And we are talking about genetic ancestry that is consistent with deriving from a major worldwide dispersal less than 60 kilo years ago, going and can explain the various findings at these sites all the way into Eastern Asia, Eurasia, and European sites, right? Worldwide expansion and archaic admixture. Locations of early individuals with modern human ancestry in Eurasia, together with sites that may indicate an earlier dispersal in Asia and Sahul, the continental shelf centered on Australia. In B, we are looking at paleoanthropological and archaeological evidence of early modern humans far away from Africa. So going back here 100,000 years, we are looking at the Chinese caves with modern human-like remains, and coming closer to 60,000 years in these two sites here. And the uncertainty in the genetic ancestry itself is shown by the size of these gray circles. So this is timing or population topology that is uncertain here. And then we get a little bit more certain and try to track admixtures of the main Neanderthal at this site, Y chromosome diversification. I will say what that is in a minute, and we'll come back to it later in another chapter as well, and the mitochondrial DNA diversification and southern Denisovan admixture. So when human beings sexually reproduce, the Y chromosome is the male chromosome that gets mixed, so the child inherits the mixture of parental genes. So Y chromosome, there are what are called haplotypes, so groups of genes on the chromosome that get hereditarily transferred, genetically transferred. So you end up having these haplogroups that we will see a map of in a minute. And mitochondrial DNA is mainly transmitted through the matrilineal line. So you can use the mitochondrial DNA to keep going back to find, for example, a common ancestor, whereas the Y chromosome diversification basically means how different populations or haplogroups got mixed over time. Okay, so that's in a very brief explanation. So here is the different mixtures you get as you get closer to today. So basal Eurasian gene mix emerged and then stayed this way here, and then maybe mixed with the Georgian population, West Russian population, and so on more recently during the last glacial. 2% Neanderthals got mixed in already around 55 to 60,000 years ago, and southern Denisovans and Siberian Denisovans added some to these sites here. So you are now having groups that can be identified as East European, Oceanian, so Australia and that region, West Eurasian, and Basal Eurasian, which doesn't extend all the way to the present. Okay, so this just gives you a sense of how the diaspora we've been talking about also has left behind genetic signatures of the interactions between the various species of hominins. So this is an example of the Y haplogroup. So this is the global distribution of the Y haplogroups, and you can see, I won't read all the population groups that go into these, but you can guess what they are. So you get Senegalese, Ethiopians, and so on all the way. So you see that this is mostly blue color here with a small mix, and this is almost all blue, and this has got several other mixes depending on where you are, and so on. So you again get into more isolated groups here, maybe in a sense of less mixing of haplogroups of different populations. So this overall should correspond over time to the kind of migrations, diasporas that have happened. And generally the accepted agreement is that maybe the origins happened here and then diaspora happened out of there, but even though we keep talking about independent evolutions as Denisovans and Floresiensis show. So looking at phase 2 then, so this scale goes here in terms of hypotheses just in Africa replacement, which is by now rejected, expansion pulses, and long-standing Pan-African connectivity with some archaic admixture. Okay, so to read this, modern human origins and diversification in Africa in the past 300,000 years, so we are going a little bit further in time here just to see how much archaic human gene may be still carried. So here African ancestry origins can be conceptualized as a range of models, most of which cannot be rejected based on the current data. So future data, better models may evolve to add more granularity to these in terms of detail. So models along this spectrum indicated by the color gradient of the arrow include, among others, a recent complete replacement. So some gene that originated here in East Africa admixed with all the other gene pools that existed and then ended up replacing all of the genes within Africa. So that's called in-Africa replacement, which is rejected now. Expansion pulses looks at a similar concept of origin of modern human here mixing with earlier species of hominins and then ending up with a mixture of other hominins with the modern human. Maybe the archaic humans didn't leave any fingerprint here, genetically speaking, and this mixed gene pool then got out of Africa into Eurasia. This is the long-standing Pan-African connectivity, which just says that there was no modern human evolution in East Africa, that it was maybe just mixture of the hominins that existed that still continued and evolved into modern humans from 200 kilo year to 70 kilo year, developmentally, behaviorally, anatomically, and so on, including encephalization, enlargement of the brain and the complexity of the brain, expertise, tool making, and so on and so forth. Okay. East Africa is a hypothetical origin of expansion here in these hypotheses indicated in a red dot, but other regions could represent the hypothetical origins too, so these are not ruling out other possibilities. Here, complex looking figure, but we have modern anatomy and maybe uncertain about the anatomy. Maybe it was partially modern or unknown, and there is the archaic anatomy in terms of the bipedalism, opposable thumbs, and maybe even the Adam's apple and so on. So jaw structure, cranium, eyebrows, etc., etc. And here are the various sites where these evidences have been found. Western Asia and the Mediterranean, North Africa, Central and West Africa, East Africa, and South Africa. So looking here at C, then the summary of genomic estimates of the timing of the earliest population divergences among present-day human populations in the literature. So these are the papers from which this review is produced. All estimates involve a Southern African Chloe-san or a Central African forager group compared to another present-day group and have been rescaled using a mutation rate of 1.25 times 10 to the minus 8 per site per generation. This is the molecular clock approach, wherein modeling the genetic evolution, you go back in time and assume mutation rates, and a generational interval of 29 years. So lifespan approximations are involved there. The software for or analytical method used is indicated in parentheses for each study here, the model that is used. So I won't go into too much detail, but there are details like how populations coalesce genetic information or haplogroups, and then how cross-population coalescence rates changed. So these are approximating midpoint cross coalition rates in this vertical scale. Time is along here, and there is a split with gene flow, so that populations split but they carried some genes like what we are indicating, for example, here. Okay, mixing and then carrying that gene pool outside and then splitting from this population, or there is a clean split where the population didn't carry the previous genetic coalescence information. Okay, so just take it for what they are. Just saying that there are genetic methods, DNA approaches using mutation rates and generational interval, mitochondrial DNA and Y chromosome approaches to go back in time to do this. To look at phase 1, let's start again with the Homo sapien lineage, known and uncertain Neanderthals, Denisovans, known and uncertain, and archaic lineages. So you can see there are some archaic lineages that we consider, like Homo erectus, Floresiensis, Eugenesis, and so on. So in this figure there are separate archaic genetic carriers that didn't probably add, like Naledi and Eugenesis here. So again, going back to 100,000 years, this is the approximate anatomical similarity possible to possible closest relative lineage. And again, start here with Eritrea, with archaic, with some archaic genetic impression of East Asian, Homo erectus, Southeast Asian, Homo erectus, but you're starting with the Buya Eritrea signature, going into antecessor in Spain, Algeria. So you can again see the uncertainties of the population types. Reading here, separation of modern human and archaic ancestries in the past 1 million years. Did I say 100,000? That should be a million. Locations of key Homo sapiens, Neanderthal, so that's the map we looked at here. Coming to this one, chronology of archaic human populations that are unlikely to have contributed to modern human ancestry. So those are the ones that probably didn't leave any signatures. These include Homo naledi, Homo floresiensis, and Luzonensis, not Euzo, Luzonensis. These biological names are difficult for me, as you can imagine. The timeline is the same as in C here, and C shows chronology and probable ancestry history of this. So you are coming again to modern humans here. History of the separation between modern human and archaic human ancestries, selected fossils older than 80,000 years, and their possible lineages attributions are indicated as here. The placement of fossils along the vertical axis reflects our assessment of how closely related they might be to the genetic ancestries. Okay, so there are again mitochondrial DNA approaches and haplotype Y chromosome approaches, and gray circles represent the uncertainty around timing or population topology. Okay, so the same story again. Various mixes. Modern humans carry 2% Neanderthal genes, some 0.3% Southern Denisovan genes, and 0.1% Siberian Denisovan genes, and the super archaic signature was there around 300,000 years ago, but probably disappeared by now. There are other details that, depending on which population you look at in modern human beings, the percentage of relative contribution of DNA from Neanderthals and Denisovans also seems to be different. So this is kind of just something I wanted to leave here, and it is very brief and not very detailed in terms of the genetics, which obviously I'm not an expert on, but just kind of interesting information that I wanted to leave here. Okay.